coming. It's a great pleasure to introduce you Laura Castañar. Most of you already know her, but for those of you who haven't had the pleasure to meet her before, Laura Castañar, she's a research fellow at the Complutense University of Madrid, currently working on the development and application of advanced solution state NMR techniques for the analysis of molecules with chemical and biological interest. She earned her BSc in chemistry at Complutense University in 2011 and her master's on chemical science and technology at Autónoma University of Barcelona in 2012. In 2015, she finished her PhD on NMR methodology under the supervision of Dr. Theodor Parella. Then she moved to the University of Manchester to do her postdoctoral research under the supervision of Professor Gareth Morris. In 2019, she was awarded a Dummy Kathleen Oleren Shaw Fellowship. And in 2023, Atracción to carry out her research as an independent researcher in the Complutense University. Her research focuses on creating cutting-edge NMR tools for determining molecular structures and understanding their behavior, and for the qualitative and quantitative analysis of complex systems with chemical and biological interest. The major vendors of NMR hardware have included her experiments in their standard pulse sequence libraries, and many NMR laboratories currently benefit from them. Since she started her research career in 2012, she had published 35 scientific papers and has contributed to numerous national and international scientific meetings with 36 oral presentations, 31 as invited speaker, this being selected by the UK Royal Society to attend to the renowned 67th Lindau Nobel Laureate meeting. She is also a member of the Scientific Committee of PANIC and Euromar conferences and of the SMASH organizing committee. So, no more to be said. Please, Laura, welcome, and the floor is yours.
Thank you for the introduction. I don't know if you hear me. I'm going to try my best. So, thanks for the invitation. It's a great pleasure for me to be here again. This time for more scientific talk about what I've been doing over the last 10 years since I started my research career as a PhD student. The title of my talk is New NMR Methods for Studying Complex Systems. So basically, for most of you are aware, I'm working with nuclear magnetic resonance, NMR, and NMR is one of the most useful and powerful analytical tools, and it's very versatile. One of the main strengths of NMR, this goes without saying that no technique on its own normally is enough, and the combination of several techniques is normally needed to solve the chemical or biological problem we are interested. But as my focus is on NMR, I need to enhance the good points of using NMR. From an analytical point of view, the main strengths are that it's non-destructive, doesn't require the separation of the components, so we can analyze components in their intact media or intact mixtures, and it's very versatile. It's very versatile because we can manipulate spin magnetization, so we can manipulate our spins to get the information that we want. So we can get information about chemical structure, about the dynamic of the molecules, but we can also get information to identify what we have and to quantify, as NMR is intrinsically quantitative. With all that information, we can get better to know our system, and we can use that for determining the chemical properties, biological function, physical properties, and we can use it for technological applications. NMR has been applied in many different areas, both when liquid and is okay, no? When working in liquid or in solid state. The ones I'm working the most, so I'm working on the liquid state NMR side, are the use of, it's working, yes, small molecules, analysis of macromolecules, complex mixtures, and drugs, mainly. So if we think about what we normally do with NMR, the most analyzed nuclei by NMR is by far proton. There are very good reasons for that, because proton is everywhere in chemistry and biochemistry, is one of the most sensitive nuclei in NMR, because as I highlighted before, the strength of NMR, the main issue, problem, drawback is sensitivity compared with other analytical techniques. But proton, from all the active nuclei, is one of the most sensitive ones. And also is very rich in structural information. We can get a lot of information about the structures present in the sample. However, as we can see in this spectrum here, very often we encounter situations like that, where we have a lot of signal overlap in our spectrum, which reduce our spectral resolution and increase the spectral complexity. So it's difficult sometimes to extract the information we want from this kind of systems or spectra. So over the last 50 years, hundreds, if not thousands, of different NMR methods have been developed to try to solve this overlapping problem in proton. The most common ones are either using other nuclei, like carbon, fluorine, or proton, because they are less abundant, chemically abundant in the molecule, and they their signals are wider spread in the chemical shift range. Also, the use of multidimensional NMR, where we spread the signals in several dimensions, then reducing the signal overlap. And we also, there are a lot of selective experiments where we look for the information we are interested in. We target our experiments to get very specific information. However, when we have very complex systems, some of those methods or the available methods are not enough. And nowadays, NMR, from the methodologic point of view, is focused on developing new methods to solve problems that cannot be tackled with previous methods. So we are not reinventing the wheel, we are trying to solve things that cannot be solved with the tools that we already have available. So this is what I've been doing over the last 10 years, is the development of NMR experiments or pulse sequences. So this is the only pulse sequence I'm going to show today, because I'm aware that the audience is not NMR focused or not in the method. So, but just for you to have an idea, what we do is basically we design experiments, we design how we apply radio frequency pulses in the time to extract the information we want from the sample, from the molecules. So basically we work on the computers, coding, arranging these radio frequency pulses in the time. We design the pulse sequence, we code it in a language that the console and the probe can understand. Then we measure the sample, and what we get is the NMR data, and from that we can extract spectroscopic information, chemical shifts, scalar couplings, NOE, and so on. With all this spectroscopic information is where we can get information about the molecules we are analyzing. So I like this analogy because some people struggle to understand what we do, and I like to say that we are kind of composers for NMR. So basically we compose the harmony of the melody, which the melody is the NMR spectra. We are focusing on working on those, while most people, chemists and biologists, are interested in the information that is getting out, in the melody of the music. So just a bit of me. I started here in Spain doing my PhD in Barcelona, in the University Autonomous of Barcelona in 2012. I went to France for a few months to work on the lab of Patrick Girardot. Then I moved to Manchester, and I've been there for the last 8 years. First as a postdoc for 3 years, and the last 4 years as a PI, as a research fellow there, where most of the things I'm going to show you today are related with this last part of my career. And since 2 months ago, I'm back here in Madrid. So this is an overview of what I've been doing, or we as a group, we've been doing over all these years. Basically, developing new NMR tools. And we use, it's very broad, and we use and focus in many different things, but I think we can categorize them in computational methods. So we use computational methods to try to understand the phenomenon behind the spectroscopy, but also to help us to design new experiments and to see if we can get the information we want. We focused in the application of the methods for determining molecular structure and behavior, or for analyzing mixtures, and in many cases to analyze the data we are getting from these advanced experiments. Current software are not suitable, so we also work on the development of some softwares to process this advanced data that we are working. Today, I'm not going to talk about pulse sequences and about what we do exactly when in each of the experiments, but it's more about to show you the capabilities of all those methods, why they are useful, how they can be used. So I will focus on these three applications, or these three kind of experiments: pure sieve, experiments using relaxation, and experiments using spectral editing. So let's gonna start with the first ones, pure sieve NMR. For those that you know me, I guess you know what is pure sieve. I've been doing that for a lot of time, but can you hands up the people who knows what pure sieve is? Well, half of the audience is not bad. So pure sieve NMR is something very old but relatively new, known and developed, basically over the last 12 years. So pure sieve NMR, they are a class of experiments that allows us to obtain proton spectrum. This is a conventional proton spectrum, and this is the pure sieve one. In the pure sieve spectrum, we remove all the scalar interaction, homonuclear and heteronuclear, to get only chemical sieve information, similar to what we do in, or what we get when we acquire a carbon. Main benefits of that is spectral simplification. We reduce signal overlap, and we have a single signal per chemical environment, which enhance signal resolution and easy the spectral analysis. So this is the most technical slide I have about that, because I think it's important, even though I'm not going to explain you how the experiment works. There are so many pure sieve experiments, and not all of them can be used for everything. And for that, we need to understand a bit how the experiment works, because it seems easy, but to decouple proton from protons is a very difficult task, because it's the same channel, it's the same nuclei. So the trick we found to do that is to manipulate the spins in such a way that we can separate the effect of chemical shift from the effect of scalar coupling evolution. For doing that, we use one element. It's what we call the J-refuxing element. But basically, what this element does, as this part here, is to separate all our spins into actives, and those are the ones we are going to observe, and passives. Those we are going to manipulate them, but we will not observe them. So by doing a trick like that, we can leave chemical sieve evolving while manipulating the scalar coupling evolution to remove it, the effect of it from the spectrum. This is during the pulse sequence, during the experiment, but they also evolve during the FID while we are acquiring the data. So we need an extra trick to make sure that we don't have evolution with those during the acquisition. So basically, we acquire the data in small chunks where the evolution of those scalar couplings is negligible. There are many different methods, but they are mainly classified depending on what element is used here for selecting the active spin and what acquisition scheme we are using. In terms of the active spin refocusing, if we go here, there are 4 of them available. I'm not going to get into the details of those, but from a practical point of view, there are 2 important features we need to be aware of. First one is, what are we going to get out? Are we going to get out our full spectrum or just a part of it? If we want to get all the information about a narrow part of it, we will use this band selective approach. If we want to get the full spectral window, the broad band spectrum, then we have 3 different experiments available for doing that. But we pay a price to this spectral simplification, and it's sensitivity. And this is very important with PureSIF. Many people is trying to use PureSIF nowadays without really thinking that there is a big price in sensitivity by this spectral simplification. And those are the numbers here. With the band selective, is the only one that we don't pay a price and actually we increase the sensitivity. With everything else, we are between 1, 3, 20% the maximum of the signal. So it's a big price. So we need to be aware of those differences for the specific problem we want to look at. And also, if we look at the 2 acquisition methods, there is one that we acquire the data as a pseudo 2D, one we acquired in real time. We also pay a price in time, and time is sensitivity. They are related. In the band selective, in the real time one, we don't pay any price because we acquire everything consecutively in the same way as the conventional. But in the interferogram one, we pay a price for the increments we need to acquire. You would say, then use always real time. Well, we cannot. So the ideal situation will be all our PureSIF experiments to be acquired with full sensitivity in the active spin element and in the acquisition. But that's not possible currently. Maybe in a few years' time we find a new method that allows us to do that. So it's important, the sensitivity is really important here, and the information that we want to get. So now I'm going to go through some of the experiments, but with examples of when we use them. So the first one is the very good one, the one that we don't pay any price in sensitivity, the one that we retain all the sensitivity or even gain it. And we do that only we are interested in a region of the spectrum. So these experiments are very useful for looking at a spectra or a molecules that they have a specific kind of signal in a region and they are not coupled to each other. Proteins, peptides, and so on. Here is a small peptide, the cyclosporine, and here we have the alpha region, so all the signals appearing here correspond to the alpha protons in the structure, and they are not coupled to each other, so we can easily decouple them and remove all the scalar interactions. NH part, so this is a very simple peptide, but when we are looking to protein disorder in proteins, and so we have hundreds of NH signals in that region, PureSIF can help us here to improve resolution and sensitivity. And if we look in detail here, we can see that all the scalar coupling interaction is removed, with the exception of these 2 signals here, that are with an asterisk. And this is because these 2 proton signals are coupled to each other. So if they are coupled to each other, we will not be able to remove the interaction between them if we are observing them both at the same time with this method. And this is actually these 2 protons here. So these are not the alpha protons in the molecule. But it's important to keep in mind because people don't think about it when used experiment. And normally say, no, I don't get PureSIF. Yeah, you are not using it right. So be aware of those limitations. It has been extensively used for the analysis of proteins because not only the band selective, but also using the BERT element, we can combine it with the real time to not lose any signal, to increase the resolution. Actually, we can increase, if we look here, this is the conventional one, and this is the PureSIF one. We gain resolution, but we also gain sensitivity. And this is because the HSQC experiment, we are already paying a price. We only observe 1% of the signals in the case of the carbon, or even less in the case of the nitrogen, because we only observe signals that are filtered by the isotopic effect. So here, we can perform PureSIF without really paying any price. And people in proteins, mainly nowadays in disordered proteins, they use it a lot to simplify all the NH region of the protein. In chemistry, we can use it also to look at mixtures of things that they are not coupled. So we can use it to do enantiomeric studies. So if we have a mixture of different enantiomers, Basically, enantiomers cannot be distinguished by NMR. We need to do some trick to them, so we need to add a chiral solvating agent or something. So they really create a diastereoisomer complex. Now we can resolve them, but most of the time, if we have different enantiomers, the separation is good, we will see, but they will overlap. And this is the case we have here, for example, or here in this enantiomeric mixture. When we have the normal NMR, no separation between the enantiomers. When we add the chiral solvating agents, then we can see that they separate, but the stand multiplicity and they appear a very similar chemical SIF make them overlap. When we use the pure SIF, in this case band selective real time, we can remove all the scalar interactions. We have a single peak for each environment. We can integrate, quantify the enantiomeric effect and determine them and quantify the ratio of the enantiomers present. So that's been very useful for those studies, and also when analyzing diastereomeric mixtures, because they are very similar in the structure, they commonly present signal overlap, and here is an example of those. This was a collaboration where our collaborator has a mixture of 6 different diastereoisomers in the sample, and he knew they should be all around here, but he didn't know how many. This is the signal in the conventional HSQC experiment. So there are several things we can do to increase resolution. It's not only pure SIF, but they can all be combined, and this is an example of that. Here we were combining it with spectral aliasing, basically gaining resolution in the indirect dimension of the HSQC. Then we were performing pure SIF, and then we remove all the contributions in the proton dimension, so we increase the resolution in the proton dimension, but it can be also combined with non-uniform sampling, which further increase the resolution in the indirect dimension. So at the end, what we got is this ultra-high resolved HSQC spectrum where the 6 components of the mixture could be easily identified if we compare with the original HSQC one. So it's very useful, keep in mind, always when we have problems of signal overlap. And a very common case of signal overlap are mixers. So this was one of the mixers we were analyzing. It was a natural peppermint essential oil. This is the proton spectrum. And we say, yes, let's gonna try to identify the components, quantify them, let's gonna use pure SIF. We use pure SIF, in this case broadband, so we were paying a price in sensitivity, but still this sample is highly concentrated, not much of a problem. We can acquire the pure SIF in less than 1 minute. It's okay. When we acquired the pure SIF, we were very happy to see the simplification, but still from the 1D, it's very difficult to extract information on its own for the conventional proton pure SIF. We did several tricks, we tried different things, and one of the things we tried was to use selective experiments. So could we combine pure SIF with things like selective toxi, where we target a specific proton and get information about all the spins in its spin system? So what we did here was to target different proton signals, run the selective toxis and the pure SIF one. So on the left, you can see the conventional proton and the conventional selective toxis, much more simplified than the original one, but still with a lot of signal overlap in some of the regions. When we did the pure SIF version of it, we could extract all the chemical SIF. We had only one signal per chemical environment, and we were able to identify and quantify the main components of the mixture, which in this case was menthol, neomenthol, and menthol. Another kind of mixers we are very interested, because we have some collaboration with pharmaceutical companies, is fluorinated mixers. Fluorination of drugs is very common and very fashion nowadays because it's increased their pharmacological activity, but very often they present themselves as mixtures. And this is a further complication because now we have present another nuclei, fluorine, that is active, highly abundant, 100%, spin half. So any complication we had before from the proton coupling now just increased by the presence of that fluorine in the system. So we were analyzing these fluoroproline systems that they had. They have a mixture of all those conformers in the sample. And this is the proton spectrum. It was a lot of signal overlap. They are very similar. They all resonate in the same region. We did PureShift, and we were very happy. Yes, much more simplified. But when we were looking at the PureShift spectrum, this is the conventional PureShift, what everyone is using until last year. When we were looking at that, we say, okay, we have proton chemical sieve information. We don't have proton-proton couplings, but PureShift conventional methods, they don't deal with heteronuclear couplings. So any heteronuclear coupling that is present is still present in the conventional methods. So the question here is, how do we know what is intrinsically a chemical sieve or what is the splitting due to the heteronuclear coupling? In some cases, we could guess and say, like for example here, and this is not as easy as just decoupling during the acquisition. We cannot do that. We get a mess. So we work on the development of a PureShift experiment that is general and can deal with that, and this is something we published last year, well, December, and it's what we call fully PureShift experiment. So in these experiments, now we add several pulses in the sequence, and now we are able to remove all the scalar contributions, not only from the protons, but from any heteronuclear heteronuclei. So now we have a really fully PureShift spectrum containing only chemical sieve information, and if we want to get information about the heteronuclear couplings, it's very easy because we just need to compare those two. We don't have any proton-proton coupling. All the extra splitting is due to the heteronuclear, so we can extract them relatively, well, quite easy. We also use it a lot to measure residual dipolar couplings, which is a very useful tool to use for structural analysis in NMR. We could measure at the same time the coupling without the aligning media, the coupling with the aligning media in the same spectrum. As we don't have any proton-proton coupling, we can extract these heteronuclear RDCs, which are key for the input normally of in the molecular calculation of the structure. The same way, if we remove all the homonuclear ones and only the heteronuclears are present, we can measure them. For the one bond heteronuclear couplings, mainly with carbon, that's relatively easy. They are 150 Hertz, so they are big. But if we are interested in long-range couplings, which they are really small, they are normally at the same size of the proton-proton or even smaller, between 2 and 5 Hertz. It's very tricky. We have all the protons there. So by removing, this is the conventional one where we have all of them. This is a very simple case for you to see. But if we remove all the proton-proton and only the heteronuclears remain, we can extract all those and combining with other techniques like IPAP, so we can measure couplings as small as 0.5, 1 Hertz, and get information, long-range information about the conformation of the structure. Since I went to Manchester, they work a lot with diffusion NMR. Diffusion NMR can be used for getting information about the size of the molecules. It can be used to get information about distribution of molecular size, in polymeric size, but it can be used as well as a way to analyze mixtures. If you have components that they diffuse differently, you can separate them based on their diffusion behavior. So they work a lot with that, and we were working on a mixture of vitamin D and its precursor, provitamin D, and we had a big problem with these ones because they are structurally very similar, and anything we were doing, they were basically overlapping all the time. Overlapping the proton dimension, overlapping the diffusion dimension, their diffusion coefficient is very similar. We give it a try to see if we remove part or all the problems of overlapping in the proton dimension, if we were able to differentiate them, extract them, and assign them. And that's what we did with PureShift. So here is an extraction of the PureShift proton dimension. So as you can see here, it's much more simplified than this one. All the homonuclear couplings are gone. And what we get is clearly aligned a set of signals corresponding to the vitamin D and the provitamin D. We were able to measure the diffusion coefficient, and those two diffusion coefficients are very, very similar. It's about 4% difference between them. No way that with the conventional diffusion experiment are going to be able to separate those. So we were very happy to see that PureSIF can be also used to increase resolution of the diffusion experiments. And then I'm going to move now to relaxation. PureSIF may jump in a few times. And relaxation NMR, everyone use it, everyone measure it. It gives us a lot of information about the dynamics, information about the bindings. It gives us information about rotations of angles and conformations. And we were interested to see if PureSIF could help to measure those values when we have signal overlap. So the first thing we did was to implement the most sensitive one, the band selective one, real time, in the conventional experiments to measure T1 and T2 relaxation rates. So we successfully implemented in inversion recovery experiments. So this one here, we were able to measure the individual T1s of an overlapping region. T2, we implemented it in CPMG. We know many people is using CPMG as the standard method to measure T2. However, mainly with small molecules where we can really see all the pattern of the spectra, modulation is a problem. So project gives much better results to measure T2. So we implemented it in project. However, I say we work a lot with T2. And then we were trying to compare different methods to measure T2 relaxation rates. And our surprise was that we were not getting the same T2 value with all the methods. Naively, you would think T2 is an intrinsic property, it should be the same, no matter what you measure it with. But it's not, because the different experiments do different things, and the answer we are getting is different. So this paper is a bit controversial, because basically we are saying to the scientific community, be careful. Be careful what experiment you use to measure T2, and what are you going to do with the T2 value after all. If you just want differentiation, it's okay. But if you want to use your T2 for quantitation, to quantify the binding or to quantify behavior, be very careful. Because in this example, here as a test sample, the cyclosporine, the blue bars correspond to project, the red to CPMG. Those are the most used methods. You can see that they do not give the same T2s. In some cases, there is a big difference between them. And then we start thinking about it and looking at the experiment and say, yes, that's because the experiment works in a different way. Both of them, they mix the relaxation, well, they mix coherencies between the two coupled spins. So if you don't have any coupled spin, no problem. You have a singlet. As soon as you have something coupled, the other spin relaxation, the relaxation of the other spin is affecting the one you are measuring. And this effect is different depending on the experiment. So we try to design a new experiment that we call it true, that get rid of this mixing, so we can measure the real or the intrinsic T2 without the effect of the other nuclei. So for you to have an idea, we did some simulations on a very, very basic spin system. Two spins, not strongly coupled, weakly coupled, A and X. If there is no coupling between them, and we use these methods, the three methods to measure T2s, we were getting the same results. The black one here is the real one. So this is a simulation using spinach. So we say, we know we have a T2 of 0.5 seconds, wherever. So the black one is the reference number. If we don't have any coupling, everything works nicely. As soon as we have any coupling in the spin, every experiment gives you a different result. Both of them, the commonly used one, the CPMG and the project, give you more T2 that is the real one. And in the case of the true, what we are doing with the true is we apply the same idea of the PureSIF, so we separate the spins into active and passive. So basically the one that we are detecting is not seeing the other one. So the influence of the relaxation of the coupled one is not affecting the one we are measuring. Only if we do that, we can get a value that is the one that we are inputting. That's not a problem if we want to use relaxation for differentiation. But if we want to use different T2 values for quantification, that's very important. And we proved that in the paper by looking at the calculation of the activation energy of this amide rotation. We were getting values with all of them, but only when we were using the new true experiment, the T2 value, the activation energy obtained from this calculation is the one we should be obtaining. So we compare it with other methods not using NMR. So just be careful how you use your T2s when you measure them. And while we were working with all this relaxation, we were thinking, nice, so we can use relaxation to quantify interactions, to rotations, but can we use relaxation to analyze mixers? Because very often we encounter the problem that the diffusion coefficient of the molecules or the components of the molecules is exactly the same. So even if we do pure sieve here, if they have the same diffusion coefficient, we are not going to be able to separate them. We thought maybe we do some relaxation experiment. That we do. So we try to acquire some relaxation data. This is the 2D plot of a T2 experiment of the project. And then we say, in principle, no, because T2 is not is an intrinsic value for each spin. It's not the same for the whole molecule. Each different spin has a different T2 value. So we can see here we have spread values of T2s depending on the proton. However, we thought, can we do a trick? So this is what we did in this experiment. We call it REST. And we say, okay, so what happens if we encode the relaxation? Each different spin is going to have its own relaxation. Now we select one. So we are going to target it one of the spins, normally anomeric or alpha protons or things like that. Can we transfer that relaxation information to all the protons or all the spins in its spin system? Because now all the protons that are in the same spin system as the selected one, they do not display their own relaxation time, but the one of the selected one, because we encode it, transfer, and measure. So by doing that, now we have all those signals encoded by just one relaxation time. We did that here, we repeat it, and we use it quite a lot for the analysis of sugars. And we look here at the anomerics. So when we can see here in the anomerics is that these two, they have a slightly different relaxation time. They can give us information about the whole spin system here. So if we use this experiment, now we can separate and get two lines, two subspectra containing only the information of those two. So since then, we realized we can use relaxation for mixer analysis. We just need to do some tricks and need to play a bit with it. Of course, we test our method as much as we could with different samples, and whenever you think it's working, try somewhere else, a bit more complicated, and it fails. In this case, we try the rest. We were expecting to see nice 3 signals here. We have the 3 anomerics, but we still have a lot of signal overlap. And this is because, remember, in relaxation as in diffusion, this is not a 2D Fourier transform experiment. This is a statistical fitting. We are fitting our data for an exponential decay. If we have several components overlapping in one of the dimensions, in the proton dimension, we are going to see an average. We are not going to be able to differentiate them. So the only way we can really separate them is if they do not overlap. So we implemented here the pure sieve approach, removing scalar couplings, reduce signal overlap. By doing that, now we can use these experiments where we can nicely separate the 3 components of the mixers. If we go for even more complicated mixers, we were analyzing a beer sample with lots of sugar in it. It also fails. So we need to go a step further. And what we did here was to do a multivariate analysis where instead of having just 2 variables that we had before, we have just relaxation and chemical sieve information. Now we have a third component, a third variable. In this case is what we call T1 toxi encoding. So it has to be something that is not dependent on the other 2, that the information is orthogonal, so we can do the mathematical analysis and analyze this data as a trigonal data. We need to design experiments specifically to get this orthogonal data. So we did that in the experiment we call scalpel, sorry, and we were able from this spectra to extract the individual components present there. The three orthogonal measurement or identities were chemical shift, this is spectral mode, diffusion in that case, so we were measuring diffusion, and this T1 and toxic encoding. This one doesn't really give us any information. It's just a way of differentiating different molecules. This one gives us spectroscopic chemical shift information, and also we can measure J couplings, and so this one gives us diffusion information. But it was the first time we were able to separate things that they were virtually exactly the same in chemical shape and the same in the diffusion dimension. So those experiments are very useful for that. As you can see, this data is the extract data. We measure the experiments and we need to extract all this data. Conventional softwares cannot do that. So that's why one of the softwares we developed is SNAT, and this is the little symbol of the software, allows you to import this data and give you these three modes where you can see spectral information, diffusion information, or if you want, just to see the toxic, although it doesn't really give us any spectroscopy information. So the last part is about methods for doing spectral editing, because I've been talking a lot of using selective pulses to target one specific signal and get information of protons or nuclei related with that signal to try to simplify the analysis. One of the methods we were working, again, was we are working in these fluorine mixers. We were trying to see if we could virtually separate the components of the mixer by using the benefit of both proton and fluorine. So this is an example of a drug mixer that we were getting from our collaborator, AstraZeneca. The 4 fluorinated mixers, proton spectrum is nice, but still a lot of signal overlap. When you do the 2Ds, it's very difficult really to assign them, and mainly the ones that are the minor components. Fluorine NMR, very nice, very few signals, high resolution. But here we have resolution, but we do not have much structural information. We cannot really assemble the structure. All the structural information is encoded in the proton dimension. So we thought, can we get the benefit of those two in a fast way? So this is what we do normally in 2D NMR. We get correlations between heteronuclei, but they can be very time consuming, and for this kind of system can take hours. Well, people you work with proteins, hours for you is nothing, but for us, more than 10 minutes is a long experiment. So we were trying to do something that is more target, fast, you can get it in a few seconds. So we were thinking, can we take advantage of the high resolution here to obtain information from here, because this is the information we want, structural information. So this is the idea behind what we call FESTA, fluorine edited experiments. So basically what we are doing, if we have any fluorine in our system, we take the advantage of the high resolution in fluorine. We can easily select one fluorine. We transfer magnetization from fluorine, sorry, go back, no, forward. We can transfer information from fluorine to any proton that is coupled to it via scalar coupling, in this case proton 6, and then we can spread that magnetization through all the spin system of the proton. So now we can get what we call subspectra from the spectrum. So this is a much simplified spectra containing only information about all the proton spin system related with the fluorine. This is a scalar. We could also exploit that, and we are actually working on it through NOE interactions for bigger things. But it allows us to obtain something simpler that now we can try to extract useful information from it. In that case, it was a collaboration with AstraZeneca. That was a mixer they were interested, and we were able to run the different FESTA, selecting different fluorines here, and getting all the different proton subspectra and do the assignment of all the major components and minor components of the mixture. And the last part is about a method that is very new, but it has caught a lot of attention. So first time we published this method was 2021. It's called gemstone, and it's a method targeted to selectively excite the spins that we want. And you will say, yeah, you've been talking all that time about selecting things with no problem. Yes, because, and it's something to keep in mind, conventional methods, we can use them, selective conventional methods, we can use them if our signals are isolated. So in something like that, we could easily select all those signals. They are separated, no overlap. We can select one signal and it's only going to be one. What do we do if we want information from something that is there? How do we get it? If we acquire the conventional selective methods, we try to use a very, very, very selective pools to select just the frequency we want, we fail in something like that. So here we try, and we knew the information about the chemical sieve because we ran the pure sieve experiment. Now we don't have proton-proton couplings, and we can extract the exact chemical sieve of all our signals. So if we run different selective experiments on that exact chemical sieve with conventional methods, we kind of get it, but multiplicity is distorted, not full signal is get, and other signals are also excited. There is one method viable that is an ultra-selective method. Well, this is just 1 minute, but we don't get the information we want. There is a method viable that is an ultra-selective method that is the chemical sieve filter. And this is very useful because now we are able to extract each of the signals, so select only the signal we are interested in. This is normally the starting point of something else. We are normally not interested just in one signal. Multiplicity is retained, nothing else is excited, but it needs to be acquired in a pseudo 2D mode. So it requires time. And if you want to implement that in a 2D experiment, it automatically converts it in a 3D, which exponentially increases the time. So the new methodology we call gemstone is able to do the same as the chemical filter, but in a single shot. Now we don't need to acquire a pseudo 2D. So we are able to select this. Select the signals we want, retain multiplicity, but in the same experimental time as the conventional one. So, as I said, gemstone on its own, well, you may be very interested in one single signal, but on its own is not very interested. So what we did over the last year and a half was to try to implement this building block in conventional selective methods. And we successfully implemented it in COSI, so we can get now selective toxins where, despite the signal being completely overlapped in the proton, this is the conventional method, we get a mix of everything. Now we can separate each of the individual components and the COSI system, so we get the correlations from them. We extend it to toxi, which sometimes is much more useful here for a study of flavonoids present in the citric. And we, again, this is the target signal, everything overlaps here, 4 different components here. We could easily select and extract information of all of them to do the assignment. We also implemented it in NOSI, which normally is very useful. Selective NOSIs are very useful experiment because normally you use NOE to define your 3-dimensional structure once you already have most of the information of the molecule. So it's much more efficient to look for the one you are interested in to acquire the full 2D. It depends how many signals do you need, but in our case, in most cases, medium-sized molecules, selective ones are much more effective. So we implemented also in the gemstone, so in the NOSI, and we were able to extract all the unique correlation for signals that overlap. This is the conventional selective NOSI. There's no way we know which one is correlated through the space with the one that is here, because there are 3. And this year, I don't remember, very recently, we applied to ROE as well, which is a bit trickier because the purity of the ROE experiment is not great. Normally we get a lot of signal artifacts, and getting this to work was complicated, and more because here we were doing a collaboration and we were looking at a biological sample. We were looking at this hexasaccharide present in human milk. Concentration was extremely low, but we were able to do the ROE measurement of the signal. So here, this was the targeted signal. Initially we thought it was a triplet, but no, we ran the pure sieve. We saw it was 2 signals there, so they belong to 2 different signals. We ran the gemstone ROSI on all of them, and we could get all the through space interactions to assign not only inter but intra ring correlations. Just a quick mention on the softwares. The most general one is the NAT. It's general, it can process any data, it's free, it's available for you to download and to play with it. It's run in MATLAB, but if you don't have MATLAB, there is also an interface that you can run it without MATLAB. It's a package. An extension of that also is to process 3D data, mainly diffusion data, because none of the available software can deal with three-dimensional diffusional data. And if you are interested on any of those samples, you can give it a try for yourself. Everything we publish, we make it freely available in the website. So those are the websites of the two groups I've been working so far, where I did my PhD in Barcelona and the one in Manchester. All pull sequences, but we know it's not easy to implement pull sequences. So what we normally do is we provide a data set, so a folder that contains a test sample with parameters, pull sequence also. People can take that, copy directly that in their spectrometers, make sure everything is correct, and from that start. The software is also available. We are very happy when someone try to do something and fails and then contact us, not because they fail, but because they contact us. So it means they are interested, they are trying, and we really help them. So either we connect to their spectrometers, or we go visit, or we give them guidance. So don't worry if you try and fail. If you don't want to try yourself, very happy to collaborate. If you have problems that you think you cannot solve by NMR, talk to people like me, methodologists, because maybe we can design something that it works for you. And that's the take-home message. NMR is very alive. There are many, many different things we can still do, and I don't see where is the limit. There is a problem. Can we tackle it? Can we design something to focus on that problem? So if you have those kind of problems, please do not give up. I mean, use other techniques, complementary techniques, that's completely fine. But don't give up in NMR just because the methods you know do not give you what you want, because there is something we can do. And maybe there are some methods there that already can give you some information. So I think that's all. As you can see, it was a lot of work here, so I haven't done those on my own, never. So I think it needs a big thanks to everyone that has been involved in all this work over the last 10 years, people from Barcelona, mainly my supervisor, Theodore Pareja and Beth Virgili, from Manchester, Professor Gareth Morris, Matthias Nilsson, and Ralph Adam, and all our collaborators over those years. And special thanks to the people who is giving us the money to develop and to work on that. So needs to be thanks, and of course thank you all for your attention.
Hello. Yeah. Thank you for this. I'm gonna stay here. For this very nice presentation. Following your analogy, I've seen that you've been a very prolific composer and have released a lot of songs and albums. So thank you for this very nice presentation. I guess that we have questions from the NMR group.
Or non-NMR, eh?
Well, thank you very much. I've been following all your talks, and they are all awesome. So I always learn something new. I was wondering about the band selective method. The limiting part is because of the number of peaks, or just because of the distance in the chemical shift, or?
So the thing is, as you need to manipulate the couple of spins. So let me see. Maybe as you need to. Oh, that's gonna be terrible. As you need to manipulate the active and the passive spins, the thing of the band selective is that it only works if the signals you are observing are not coupled within them. So it doesn't matter if it's 1 ppm, 20 ppm, but if those signals are not coupled within them, so for example, the alpha protons, they do not couple to each other, all their coupled partners and someone else in the spectrum, that's okay. So you can use it for targeting 10 hertz, 100 hertz, or 1,000 hertz. It doesn't really matter. There is not a limitation on that. But if there are signals that are coupled with each other within the bandwidth of the selective pulse, there is no way you can remove the effect of the coupling between them. Some people has used that because what you remove is the coupling to everything else. So if you have a very complicated multiplet, you end up with a doublet because you only have one partner coupled there. So you only get the coupling with that individual one. So actually people is using that to reintroduce particular couplings. But there is no limitation on that sense. As just need to be aware, if there are things that are coupled in that region, band selective is not your option. Has to go for Sanger-Stern or a PSIC or a PERT.
Questions?
So much for this fantastic talk. I was curious. I'm not sure. Oh, sorry.
Mm-hmm. Yeah, so first question. Regarding the aromatics, we've been doing quite a lot of work on purity on aromatics. Everything that is strongly coupled is problematic, no matter what experiment you use. But yes, you can decouple strongly coupled aromatic systems, mainly with HSQC, because each of those protons is coupled normally to a different carbon-13. That's a different thing to keep in mind for people who does proteins. Many people try to read those papers, and we use HSQC, BERT, PureShift to get rid of things there. But this only works if we don't have fully labeled molecules, because we rely on the isotopic labeling being very dilute to not having things coupled to each other. But if all your carbon are carbon-13, then all your protons are going to be coupled with the same carbon. It's going to drastically fail. So you will need to use other methods. It's not that you cannot do it, but you cannot use PERT. But yes, but you have that problem with the aromatic. It will work. You will focus on that region. That's the other thing. We know the techniques very well. I will not run something general for that. I will target for that region. I will use either a region specific or a isotopic labeling specific for that problem. But yes, it can be done. Regarding the biosolids, solid NMR is a different word. Dipolar couplings are massively overwhelming. Any scalar coupling, you don't see them there. Decoupling those dipolar couplings is a different story. Some preliminary initial work has been done on that. So one of our PhD students, Penelope Motsuri, went to work with Lyndon Ensley and transitioned from liquid to solid and tried to implement this idea of PureShift in solid. And she has published a couple of papers on that. So I think people in solid are starting to think, can we use those ideas to simplify ours? And I think we can, but it's everything to be done. Because so far, this has been mainly applying liquids. So yeah, I'm looking for your new toy to arrive now that I'm here. And I think there are many things we could try and see how far we can push it to increase the resolution there.
Thank you.
Any more questions from the audience?
nice talk. I wonder when you powerful that you can analyze different mixtures and how these mixtures, how what's the level of complexity of these mixtures you can handle, because you're competing with methods like, for instance, mass spec, which are very powerful. Instead, you have the information of structural information, and it's more more rich, the information, but where are the limits in the terms of concentration, number of compounds, I would say difference in concentrations?
Yeah, I think that's a very nice question because it makes us rethink, not like think carefully about what we do. We start doing research on techniques and we specialize so much in them that we forget that all the other analytical techniques exist. We should not do that. We should all work together and they are all complementary. So as you well say, mass spec on those ones is great. Sensitivity is great. So actually when we are working now, we have a couple of projects on metabonomic projects, and the complexity of the spectrum is massive. We have hundreds of components in it. From the NMR perspective, it's not a problem. I mean, it doesn't really matter if we have 10 to 2,000. It's more complex, but we can design and target different things. The main problem of NMR is sensitivity. So actually what we are doing in those projects is we are collaborating with people working in mass spec. So it's a combined project where we get from mass spec what mass spec is really good at, telling us concentrations because the sensitivity is great. NMR is poor in sensitivity compared with mass spec, but we can get information about specific ones. So actually we need to combine them. Not a single technique on its own for those complex systems is going to give you the information you want. We need to learn to work together, because we are so used to say my technique is really, really good that we forget that the other one is also really, really good for a different purpose. So let's gonna join the strengths of them, and only in that way we are gonna get information, the full picture of what we want. So specifically answer it about NMR. Complexity is not a problem. I mean, it's not a limitation. Number of components is not a limitation. Concentration it is. So we are talking about picomolar. We struggle. So we will need to go to high fields, cryoprobes, and things like that. So yes, the amount, the main limitation of NMR, anything we do by NMR is concentration, I would say. And people you work with proteins and things like that, you're more aware of that limitation.
Sorry for that. A couple of questions. The first one being like more regarding if you could like elaborate a little bit more on the different applications from your perspective, no? Of how would your method be more like give a better advantage? Because I guess the from the last part of the last question, so I guess that your method is very nice at detecting some particular molecule from a mixture, but it has to be in a relative high amount, right? But in terms of, for instance, metabolomics was one particular question, but what could you say about applying this to, for example, imaging magnetic resonance? Would it be something that you would?
They already use it. So they use these spatial encoding techniques in magnetic resonance imaging for doing measurements on cells and things like that. So they already use it. Well, I don't know. It's difficult for me to answer that question because I don't see those methods are mine, and I don't see those methods as the only methods I do. So I think it's a constant evolution. So yes, I did that, and that can be useful for something. If there is a problem, I will do it. I will do something different to solve that problem. So in terms of metabonomics, for example, PureSieve is being, so the development of PureSIF started like 12 years ago, something like that. It has taken a while for the community to get informed and to see the benefits of them. And I would say that now, over the last couple of years, people is starting to use it. And one area that is been investigating, but is in the early days, is metabonomics and all the medical sides. Because what now people is trying to see is if this spectral simplification helps on the classification, if you are just looking at the fingerprint. So some people is not interested in the structure, is interested in the pattern, and if it's any biomarker that you can get from the pattern. And the thing is this simplification helping you to identify those biomarkers? The next step would be which one is that biomarker, which one is that molecule, what is the structure, because this could help you to target the biological metabolism of it. But there are so many different things, and the good thing about working with method development is that you are open to everything. I mean, at least I am open to investigate everything that it could be exploited. So I think the implementation is always the same for us. We develop a lot of tools, but if nobody uses them, they are useless. So we need a community to start using them to see what are the limitations or what are the limits of those, and what else do we need to do for specific cases. And I think in many of those experiments that are relatively new, so 10 years from us from the development to the implementation in the real scientific community, not NMR spectroscopies, is normally the time. We need time. We need people start using it and encounter different problems we haven't seen, or asking different scientific questions that we don't have, because we just developed the method. Most of the time we don't have the problem. But everything has limitations, and the good things for me, I'm very happy when someone tells me, I tried that, it works really nice here, but when I try there, it fails. Because for me, it opens the door to say why it fails. Can we do something else? Do we need something? Is an intrinsic limitation, which most of the time is not, if it's not concentration. Everything else, it opens the door for investigation and keep looking for trying to solve that problem. So for me, it's fascinating that people is using and have telling me your method fails, because it means I can still do things. There is something else I can do. So I don't know. We need time for see where the limitation of all those methods are and what else we can.
Okay. And the other question, like more NMR related. So have you tried, or would it be possible to couple or to combine your method with any source of paramagnetic NMR, for instance?
We are working on that. Yes. The answer is yes.
Because I guess that it would be very nice if you are able to get rid or to define which are the relaxation enhancement from the coupled spin
Actually,
systems.
that's part of the project with this fellowship. It was looked into those kind of things. Definitely, if you can quantify the enhancement by the presence of the paramagnetic nuclei there, can you get any extra quantitative information from that paramagnetic enhancement? Can you measure? Can you combine with different things? So I think there's a lot of things to do on that side, and I'm lucky now that my colleague Ángeles Canales works quite a lot with paramagnetic, and now we are going to exploit the application of all those methodologies, how can be used to enhance the information you get from paramagnetic NMR, because I think it can be very helpful, mainly for biological systems where the complexity is high.
Nice, thank you.
Read my mind.
So, okay, some more questions.
I have two questions. One is if you are tagging with in the case of the anomeric proton, you are tagging all the sugar. How big can be the sugar? You can tag all the atoms in the molecule, if it's, for example, I don't know, maybe a polymer or a oligomer. Or
So, let's see. Polymer oligomers are different to sugar. Polymers, in general, everything is within the same spin system. So, for example, by using this toxin approach, you could get information about, well, it depends how long you can be mixing magnetization to get information about the whole system. But in principle, everything that is connected by scalar coupling, you could. Sugars is different because protons from different sugar rings are not scalar coupled between them. So there is not a scalar coupling between the first ring, the second ring, and the third ring. So if you use the Toxi approach, you will only reach the information within the same spin system, normally just one. But the idea, and this is something we are working on, is can we relay information? So can we jump? So we first start from one. Can we do NOE? Can we do any kind of correlation that links one with the other? So now, for example, with the hexasaccharide, we could do relay steps. So we could start with Toxi, scalar relay, and then do NOE from there and jump to another ring, and then to relay a scalar from them, and so on. The problem there, and we need to look at it, is how far our magnetization is going to survive to be playing with it forever. Normally you will need, if you want to do something involving relay steps with NOE to jump between units that are not scalarly correlated, you will need to have a medium that enhance the dipolar interactions and that you can really have those dipolar strong enough to be used. So like viscous solvents or things like that. And this is something we are exploring. So this is the first step. Can we jump? Can we combine? Actually, the idea is these are versatile. So we could start with a scalar and then relay on NOE, but we could start with NOE and relay on a scalar. So we can jump directly to another region and expand in that region. So, and it could be different steps.
Okay. The second question is related with the mixture separation, but taking into account that if you can implement all these techniques in more than one dimension, or is it too time consuming or something like that?
No, you can. I mean, it depends what technique, it depends on the particularity of the experiments, but you could, I mean, there are dozens, if not hundreds, of 3D diffusion experiments. So DOSI is coupled with HSQC, with Toxi, so it can be, all those things can be coupled. PureShift have been implemented in 2D and 3D. Selective methods have been implemented in 2D and 3D. So yes, it can be coupled. The problem, anytime you do something in a dimension and you increase the dimensionality, you exponentially increase the experiment time. So it depends. So if it's something that it doesn't increase dimensionality, the price to pay is less because you don't need that extra time. If, for example, you do PureShift, you may pay the price of sensitivity, not with the band selective. It really depends.
Yeah, well, we combine it with something like NUS, for example.
Yes.
Maybe you can.
That's completely fine. You only gain there. So that's why band selective or HSQC real-time experiment have been very useful for 2D because you implement them and you don't pay any price. Full sensitivity, more resolution, even increase. Actually, people in proteins, for example, when they are looking at the NH region, they don't have much multiplicity. They are mainly doublets and normally broad. They even don't see the doublet there of the coupling.
I was thinking in mixtures of proteins precisely.
So what you get there, if you implement those, is no pay, extra pay in sensitivity. You gain resolution. So you didn't see the doublet before, but if you remove it, you collapse. So you gain resolution and you increase sensitivity. So actually people in proteins, when using, they are not interested in really in any coupling. They are just interested in getting more signal, as much signal as possible, as much result as possible from those broad signals. And it also helps. So, for example, now we are working, it's not proteins, but we are working in fluoropolymers. Fluoropolymers, they have lots of fluorine-fluorine couplings between them, and you look at the spectrum, it's just like a bump, 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 bump. They are not interested in the couplings, but when we implemented purity, we reduced the line width of the signals from 150 hertz to 2. And then you gain massive gain in resolution. If you have several signals there, they are just very impressed. So we did that in a collaboration. We are putting the paper together now. It's like you go from bumps to very narrow signals. They didn't have much interest at the beginning on that because they're like, yeah, we know it's just a bump involving 7 fluorides, but now they can see the exact 7 fluorides.
Yeah, it's like a magic angle technique.
So yeah, and then it's the same in proteins. So you see bumps, 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 but and you know I have 110 or 120 NH in that region. That's what you are really interested. You don't care much about the purity, but can I get a bit more of signal and a bit more of resolution? That's what people is using it for. So people is using it for their own purposes. It's not always the same. In small molecule, it's more about the characterization and getting the assignment and the correct structure from the drug. In big molecules, it's also for the assignment, but mainly sensitivity and resolution.
Last call. No. So let's finish. Thank you for the discussion. Let's thank again Laura. And finally, we have like a tradition here, appreciation. We would like to give you a souvenir from the old institute. Thank you.